So we're going to go through quite a bit in this one. I know the last couple have been super short, so hang in there. We've got a bit to get through. So by this one, we're going to talk about hydrolysis of proteins, which we mentioned earlier, and we're going to then how we might use this to identify them. So these are things you need to be able to explain, and you want to compare the use of chromatography and electrophoresis at the end. Now, this one here is a picture of electrophoresis. It's of DNA, but it's roughly the same thing. Now, hydrolysis is the chemical decomposition in which a compound is split into other compounds by reacting with water. And here we have a protein, a peptide bond, being broken down. So you can use it, to be honest, you can use it to make new proteins if you want, or you can use it to break a protein down into its constituent amino acids. And it will break it down one at a time, generally. So hydrolysis of peptide bonds. It's basically the cleavage of it, and mixing water with peptide bonds will rarely cause it to just happen on its own. It will happen, but extremely slowly, so they'll decay in water. Your peptides will decay, but it increases by a factor of 1014 when we use catalysts. So these are enzymes. So we use enzymes to do it. The one that happens in your mouth is called amylase, and it breaks down starches in your mouth. And if you've ever chewed on bread long enough that it turns sweet, that's because the amylase has broken down the amylose. The catalyst has done it in your mouth, in your saliva, and turned it from amylose to glucoses. Okay, so protein plus water gives amino acids. So consequently, the opposite's true. Amino acids, when we add them together, gives us proteins plus water, and that's why they're condensation polymers. So in humans and other mammals, hydrolysis occurs in the digestive system, in particular the stomach, where it's very acidic, as well as in other forms of chemical digestion in the mouth and on the way down and so forth. The lock and key theory, which we'll have a look at in a second, it's what makes it much more efficient. So the substrate, which is your protein, so substrate's what's being acted on. It fits into a specific place like an enzyme, and it gives it it just holds it still basically for the chemical reaction to occur. We can do this in the lab. That's actually not the big deal, and we can actually control it really well in the lab these days. And so we can break it into individual amino acids or smaller length proteins. And here's the lock and key model. So you've got your enzyme, it fits in, your substrate, it's your enzyme. This is your substrate, or the protein that's being broken apart. It fits there, and while it's sitting there, it just makes it easier for it to react, so the water will come along and cause it to hydrolyze and break apart. Now, we're going to do this in the lab, and this is roughly how we will do it. We won't do it for 24 hours, but we will reflux it with 6 molar hydrochloric acids, that's 6 mol per liter hydrochloric acids, and it will break down the peptide link. Then you neutralize it, so you stop the reaction. You would add 6 molar sodium hydroxide, for example, to neutralize it. Then use one of the two processes that we're going to talk about. And this is either separate it with paper chromatography and then spraying it with anhydride, or using electrophoresis. So we're going to spend the rest of our lesson comparing those two ideas. Chromatography and electrophoresis, they're actually both used by forensic chemists. Now, and they can both be used for protein or amino acid analysis. All right, so chromatography. Basically, you've got a stationary phase, which is a wet piece of absorbent paper. We'll use filter paper. Water gets trapped in the pores of the cellulose, and the mobile phase is the solvent. Okay, so neither of these are actually the proteins and amino acids. So you draw a line where you're starting, and it will move up the line via capillary action, so vertical line, and the solvent will bring the different, it will bring the amino acids which have been broken down with it. Now, it moves up the paper by capillary action, as we said, carries the amino acids. Each amino acid in the mixture moves up at different rates, and it's relative to the solubility in the two phases and how it reacts with the cellulose. It's not as much a size. So a more polar amino acid dissolves more readily in water and less in non-polar solvents, and will move up the paper slower. Less polar amino acids dissolve a bit less readily and in water and more in a non-polar solvent, and will move up. Now remember, we use a solvent which is less polar than water, so acetic acid quite often. And we get something like this. Now, basically the amino acids are colorless, so we add anhydride, as we talked about in the previous lesson, and it bonds with it takes the nitrogens out of the amino acids, or it uses them to make the purple chemical. The purple complex is the right way to say it. And then we use this formula here. So you got your solvent front. So you basically it creates a ratio, and each of your, depending on the solvent you're using, each amino acid moves at a different ratio, which is actually pretty cool. It's pretty consistent. So by running known amino acids on a separate chromatogram at the same conditions, much like we would do to make a what's called a calibration curve with AAS. So we run some known ones. We can identify, and it's quick and inexpensive and very, very easy to do. Then we've got electrophoresis. So electrophoresis is cool. Again, we're using porous paper or agarose gel, and we put each ends in a buffer over here with a cathode and an anode. It's connected to a high voltage power source. So for those of you doing physics, you can actually use a Van de Graaff. This is one of the things a Van de Graaff generator is for. You could use a Van de Graaff generator to power this system here. So there's our sample dead in the center, and the positively charged particles will move towards the anode, over here, and the negative charged particles move the other way. Neutral particles stay in the center. Once again, we use we set up a comparison one with known proteins to see what it does. Now, this is where size also comes into play. If you've got two proteins with the same two amino acids with the same charge, the lighter one, the smaller one, will move further along. So it's using both the size and the charge of the particles. So how do we identify it? Basically, every single amino acid has what we call an isoelectric point. So the pH which it is neutral. So positively charged at a low pH. Generally, amino acids tend to be positively charged at a low pH, and they tend to be negatively charged at a high pH. But each amino acid has its own isoelectric point that is individual. And this is essentially dependent upon the R chain. It is dependent upon the chain of the group. Up here, though, this is your one amino acid. This is what its charge is at different points. Okay, so the different R groups have different isoelectric points. If it's basic, it'll have a higher isoelectric point. If it's acidic, it'll have a lower isoelectric point. And we use those isoelectric points plus their size to differentiate between the atoms. So again, we separate it. We then put it in an oven to dry or an incubator to dry, and we spray it with anhydrous. And once we spray it with anhydrous, it goes to that purpley blue color. So if we have 3 amino acids in a mixture, we've got aspartic acid, glycine, and lysine. At pH 6, the aspartic acid is an anion and goes towards the positive terminal. The lysine is a cation and will migrate towards the negative terminal. And the glycine is a neutral deuterion at 6 and it will not move. So let's compare the two. You should be able to put this into a table. So both techniques are used for separation and analysis of amino acids. Chromatography separates by solubility in polar and non-polar solvents, while electrophoresis separates by their charge and sizes. Electrophoresis, the degree of separation, is changed by the pH of the solution and thus the charge of the amino acids. In chromatography, it's controlled by changing the polarity of the solvent. So electrophoresis is actually a lot more flexible than chromatography, and that's very, very important. One of them is much more convenient, but the other one has a higher degree of sensitivity and flexibility. So electrophoresis is more expensive than paper chromatography and requires a high voltage input. You might look at it and go, well, it costs more, so what, big deal? But when you're actually running a lab, that budgeting, that's really important, unfortunately. So positives and negatives to both, but that's a good comparison there. It's also a nice little summary. So use of electrophoresis in forensics. You can use it to identify the origin of proteins, okay? You separate it via electrophoresis. Electrophoresis is much more common than your paper chromatography, but paper chromatography is a good starting point. So different animal species have different characteristic proteins in their blood samples, and so you can identify the types of blood. Now, this was done in Australia in the early 90s with fish. So Australian restaurants were accused of replacing expensive barramundi with ling, and it turns out that that's exactly what they were doing. So, but this was done with protein electrophoresis, so that they understand what the protein profile of a ling fillet looks like, they understand what the protein profile of a barramundi fillet looks like, and then they compare the two. So it's really useful because the one that we always hear about is DNA, right? So we always hear about DNA as the one, particularly with use of electrophoresis. The problem is DNA is actually quite hard to get. It degrades very quickly, it falls apart. This is the problem with Jurassic Park. It's the only problem with Jurassic Park, otherwise that movie is perfect. And proteins, on the other hand, tend to be much more stable. So they're really useful for identifying, species. That's actually quite important. Again, if you've got organic matter there, you need to know what it comes from. However, polymorphic means there's different forms, different morphologies of certain proteins. And what's really interesting there is that we can actually, some proteins, if we can find samples of that in the blood or in the flesh or whatever, you can use it to narrow it down to groups of people. So you'll find one person, you'll get 5 or 6 polymorphic proteins. There is an example here, phosphoglucomutase. The mutase means that it's highly variable. So this is a good one for you to know. But what you do is you get 4 or 5 different proteins that are highly variable, and you compare that, and that allows you to identify an individual with a high degree of certainty. And that's the way we use it. All right, I will see you in class.